One of the most frustrating parts of aging is not simply losing muscle. It is losing the ability to rebuild it after damage. A young muscle can be injured, activate repair cells, replace damaged fibers, and recover much of its original strength. An old muscle often follows a different path. Regeneration slows, scar-like tissue accumulates, and the repaired muscle may never regain the same force. A new 2026 study suggests that part of this decline may not come from muscle stem cells themselves. It may come from the environment surrounding them. When scientists removed one protein called P311 from old mice, injured muscles regenerated better, accumulated less fibrosis, and recovered more force. That distinction matters because aging research has spent years asking how to make old muscle stem cells behave young again. But a stem cell does not work alone. After an injury, it receives instructions from immune cells, connective tissue cells, blood vessels, extracellular matrix, and chemical signals in the surrounding tissue. If that environment becomes hostile with age, even a perfectly capable repair cell may struggle to do its job. The new study published in NPJ, Aging on August 22, 2026, focused on P311, also known as NREP. P311 is a highly conserved protein previously linked to fibrosis in other tissues because it can promote production of transforming growth factor beta, or TGF-beta. That immediately made it interesting in aging muscle. TGF-beta is useful during wound healing, but too much of it can push tissue toward fibrosis. An aged skeletal muscle is already known to produce excessive TGF-beta, particularly after injury. Fibrosis is essentially a repair response that has gone too far. Imagine cutting your skin. Some scar tissue is necessary to close the wound. But skeletal muscle has a different problem. Muscle needs flexible, contractile fibers arranged so they can generate force. If too much of the damaged area is replaced by stiff connective tissue, the injury may technically heal while function remains impaired. The researchers therefore asked a simple question: what happens if you remove P311 from old muscle? They compared young mice between roughly 4 and 7 months old with old mice at 24 months. They then injured the tibialis anterior muscle, a muscle in the lower leg, using a controlled chemical injury. Some mice had normal P311. Others were genetically engineered so that P311 was absent throughout the body. The first clue came from the injury itself. In normal mice, P311 expression increased after muscle damage, but the response was even stronger in old muscle. That suggested P311 might be part of the reason the aging repair response behaves differently. Then the researchers looked at TGF-beta. Old mice lacking P311 produced less TGF-beta after injury, particularly in the aged muscle tissue. And when the researchers examined fibrosis directly, the difference became visible. Old mice without P311 developed less fibrotic tissue and showed lower expression of genes associated with fibrosis. Their muscle histology looked better than that of old normal mice after the same injury. But reducing scar tissue is only interesting if the muscle itself actually recovers. That is where the study became much stronger. The researchers measured muscle fiber regrowth and force production. Old mice lacking P311 regenerated larger muscle fibers and recovered more contractile force than old normal mice. Genetic programs involved in skeletal muscle regeneration were also more active in the old P311 deficient animals. So this was not simply a cosmetic improvement under the microscope. The repaired muscle worked better. And that leads to the central question of the study: where was P311 causing the problem? At first, you might assume it was acting inside the muscle fibers themselves, but single-cell RNA sequencing pointed somewhere else. P311 was especially abundant in fibroblasts and in a population called fibroadipogenic progenitors, or FAPs. FAPs are some of the most interesting cells in muscle aging because they can either help regeneration or make it worse. After an injury, FAPs rapidly expand. In the right environment, they support muscle stem cells and help coordinate repair. Once the job is done, many of them disappear. But in aging and chronic disease, FAPs can become excessively fibrogenic. Instead of temporarily helping rebuild the neighborhood, they begin filling it with collagen and other extracellular matrix components. Think of them as a construction crew. In young muscle, the crew arrives after damage, stabilizes the site, helps the real rebuilding teams, and then leaves. In old muscle, part of that crew may stop leaving. Instead, it keeps pouring concrete. The structure becomes more stable but less functional. The researchers isolated FAPs from aged muscle and found that removing P311 was sufficient to reduce their fibrogenic behavior. This suggested that the improvement in old muscle regeneration was driven, at least partly, by changing these support cells rather than directly modifying muscle stem cells. That may explain something important about age-related muscle decline. Old muscle stem cells are often described as defective, and some intrinsic defects absolutely do accumulate with age. But transplantation experiments and other research have repeatedly shown that the environment matters enormously. Put old cells into a more youthful environment and some functions can improve. Put young cells into a chronically inflammatory or fibrotic environment and their performance can deteriorate. The P311 study fits that broader picture. The old muscle did not necessarily need more repair cells. It needed less interference from a repair environment that had become too fibrotic. This is particularly relevant to sarcopenia. Sarcopenia is the age-related loss of muscle mass, strength, and physical function. It develops gradually and has many causes. Motor neurons decline, hormonal signals change, physical activity often decreases, mitochondrial function shifts, protein synthesis becomes less responsive, and chronic inflammation increases. But another problem is what happens when the remaining muscle is damaged. Exercise itself creates microscopic muscle damage. Falls and injuries become more common with age. Surgery can require long periods of recovery. If each repair episode becomes slightly less effective and leaves slightly more fibrosis behind, the consequences can accumulate over years. That means improving regeneration could matter even if it does not completely stop age-related muscle loss. Preserving the ability to repair may help preserve independence. This is also why muscle strength matters more than appearance. A larger muscle is not automatically a more functional muscle. What ultimately matters is whether that tissue can generate force when someone stands from a chair, climbs stairs, catches themselves during a stumble, or carries groceries. The old mice without P311 did not just show better tissue structure. They generated more force after injury. That is the kind of endpoint that makes a regeneration study more relevant to healthy aging. But there is an important limitation. These were genetically modified mice. The researchers deleted P311. There is currently no approved human drug that reproduces this intervention specifically in aging skeletal muscle. The study therefore identifies a potential therapeutic target, not a treatment that people can use. There is also a deeper challenge. P311 is not necessarily a useless protein. Biology rarely keeps a highly conserved molecule around for no reason. Its role in wound healing and tissue remodeling may be beneficial in some circumstances. Completely eliminating it throughout the body could have different consequences, from temporarily reducing its activity in aged muscle after an injury. The ideal future treatment might therefore need to target P311 selectively, or interrupt only the fibrosis-promoting part of its pathway. TGF-beta creates a similar problem. Since excessive TGF-beta contributes to fibrosis, simply blocking TGF-beta might sound attractive. But TGF-beta is involved in immune regulation, tissue repair, cell growth, and many other essential processes. Broad chronic inhibition could produce serious side effects. That is why a more upstream or cell-specific target, such as P311, is interesting. It might eventually provide a way to reduce excessive TGF-beta activity in the particular cells causing fibrosis, without shutting down the entire pathway everywhere. But that remains hypothetical. The study also used an acute chemical injury model. Human muscle aging usually develops through repeated small injuries, inactivity, metabolic disease, neurological changes, and decades of accumulated stress, rather than one controlled laboratory injury. Whether P311 plays the same role in human sarcopenia or recovery after surgery still needs to be tested. And there is another question the study cannot yet answer. Could blocking P311 improve muscle that has already become chronically fibrotic? Or does it work mainly when given before or during a new injury? Those are very different therapeutic situations. Preventing excessive scar formation is usually easier than reversing scar tissue that has been present for years. Still, the result points toward an important shift in muscle aging research. When older muscle fails to recover, the obvious instinct is to stimulate growth harder. More protein, more anabolic signals, more muscle stem cell activation. But if the surrounding tissue is becoming increasingly fibrotic, simply pushing growth may not solve the problem. Sometimes the obstacle is not that the builders are too weak. It is that the construction site has become filled with concrete. This idea also makes exercise remains one of the best supported ways to preserve muscle strength with age. But exercise depends on successful adaptation. Training stresses muscle, and the tissue must repair and remodel afterward. If aging progressively shifts that repair process toward fibrosis, understanding how to preserve a regenerative environment could eventually make exercise and rehabilitation more effective in older adults. The researchers now have a specific candidate pathway to investigate. P311 rises after muscle injury, rises even more strongly in old muscle, promotes TGF-beta-related fibrosis, and is highly expressed in the fibroblast and fat populations that shape the regenerative environment. Remove it and old muscle repairs itself more effectively. That does not mean scientists have reversed muscle aging. But they may have identified one reason old muscle becomes so bad at recovering. And that may be just as important. Because healthy aging is not about preventing every injury. It is about keeping the body capable of rebuilding itself after one happens.